over the years have abandoned the veins and worked mostly on arteries, but even in certain arteries we found the same. For example, in this cerebral artery, an artery that goes to the brain, I've shown you already experiments with that, you can see that if we look at the responses to increasing concentrations of acetylcholine, one group of preparation relaxes, another group contracts. And it is the one with this without endothelium that relaxes. And it is the one with endothelium that contracts. This is the demonstration of endothelium-dependent contraction. And again, it gives you the message that there are no general rules. You know, each blood vessel, each artery or vein has its own little world and adjusts to it for its function, its specific function. And as a consequence, these type of responses can be different. And we find that everywhere in the body. Not all muscles are the same. Not all fat cells are the same. You know, not all skin are the same. You know, skin here is much tougher than the skin here, you see. So we continuously, our tissues adjust to the function. And thus there is no general rule. You can never say in blood vessels, blah, blah, blah. You always have to say in this blood vessel, blah, blah, blah. Okay? So, here. So we had those endothelium-dependent contractions. And of course, originally we did not know what happened. And we hypothesized that maybe those endothelial cells could release a vasoconstrictor that would diffuse to the smooth muscle to cause the contraction that I just showed you. We didn't know what it was at the beginning. So, remember, we invented a nice name, endothelium-derived contracting factor, and immediately abbreviated it to EDCF, because that sounds like Vanhoutte knows what he's talking about, which was not the case. Okay? So, for several years we had this hypothesis. And today I would like to go through some of the most, and we've been following up that type of research for the last 30 years, and we still do in my lab in Hong Kong. It remains one of my major themes of research. And today I would, with you, like to show you how the process goes, how you start working and thinking about that. And I will show you a number of experiments. Now, I want to tell you that they're not necessarily in the chronological order. They are in the logical order. And the first question, if you go back to this schematic, is, does EDCF exist? So, and that question we asked for a long time. And it's only 20 years later that we had the data demonstrating that it existed. And we got data from finally succeeding in doing bioassay experiments. I've mentioned to you what bioassay studies are. If you think that two cells talk to each other, you separate them and you try to make the point. And the way we did that for EDCF, we used what we call in pharmacology the sandwich technique. Now, sandwich is not a very scientific name, so we, on this schematic, rather than saying sandwich, we use layer preparation. And the way we do that, we have a piece of vascular smooth muscle without endothelium. And that piece is connected to one of the transducers. And if you want to see how this happens, if you haven't been to the lab on the 4th floor, you know, people do that. every day. Well, not every day, almost every day. They should do it every day, but almost every day. And in this piece of tissue, we measure contractions and relaxation. And then we sandwich with it another piece of artery that has, like here shown, or does not have endothelial cells. And we bring them face to face. And actually, in this experiment, we stitch them so that the contact is very close. Now, if the presence of the endothelium makes a difference, here we measure contraction relaxation. Here we have another tissue with or without endothelium. If I see a difference between the presence or not of the endothelium, this implies that something has diffused from here to here. Otherwise you cannot explain the experiment. Do you understand that? So that's a crucial step in this experiment. Okay? And only for your attention for this part. In this experiment, the sandwich has a donor strip with endothelium. In this experiment, the sandwich has no endothelium. In the absence of endothelium, no contraction to acetylcholine. In the presence of the endothelium, contraction. This was, in 2002, more than 20 years after we hypothesized the existence of EDCF, this was the demonstration that EDCF really exists. And I was very happy about that. So yes, EDCF exists. Now, I've already tried to explain that to you. We can take blood vessels and put them in our artificial organ chambers. We can do all kinds of things. It is up to us to find responses that are physiologically relevant. For example, I mentioned that before. If we take blood vessels with endothelium and we buy a big bottle of acetylcholine, we buy it from Sigma, and then we dump acetylcholine on the blood vessel, that has no physiological significance because most of the blood vessels don't have nerves that produce acetylcholine. So you always have to ask yourself the question, is there a physiological stimulus? Remember I told you that? And the first important one that we found for EDRF responses was increases in shear stress. Well, we went along the same line, but here for this response, what we found to be physiologically relevant was stretch. And you've seen those slides already. In these experiments, we will take a ring of basilar artery and suddenly stretch it. And we will have rings with or without endothelium, and then measure the response. Remember this slide? You've seen it before. If there is no endothelium, the artery reacts as a pure elastic structure, as if it were a rubber band. We stretch it, and then through elasticity, recoil, hysteresis, some of your physics students, you know that much better than I do, we get down and we settle down at a new level of higher tension because we stretched it. But if the preparation has endothelium, rather than having this, like that here, you see that it now very strongly contracts and maintains a contraction for several minutes. Okay? That is endothelium-dependent response, and now not to a drug, but to a very simple physiological maneuver. And this stretching of a blood vessel corresponds to what every time the blood pressure increases you know, the blood vessel will be distended. You understand that? Now, remember why, when I talked about that, I told you already why I think that this is important. Does one of you remember that? Make my day. Remember. What phenomenon was that important for? Remember? Nobody? I'll show you the slide. You hopefully will remember. Autoregulation. Remember I told you that certain circulations, mainly the brain, the kidney, have to continue, you know, tissues that have to continue working irrespective of changes in blood pressure, because we start running, you know, or we get very excited, or we get very angry, you know, then the blood pressure goes up. You don't want your blood vessels to explode in the brain, so you have to protect, and you do that by autoregulation. That is, as the pressure, if the pressure increases when the blood vessel is empty, you know, progressively more blood will go through, the caliber will increase, the diameter will increase, but then there is a whole range of blood pressure where things stay the same. And that protects the circulation. And I believe that one of the key cells in that autoregulatory response are the endothelial cells. By secreting EDCF. Another question that one asks is, does this type of responses play a role in disease? Now, for endothelium-dependent relaxations, I have shown you that if we get high blood pressure, diabetes, you know, the things go downhill, so to speak. Well, the same is true for EDCF-mediated responses. But the other way around, whereas the release of NO goes down, the release of EDCF goes up progressively. And at the end of the lecture, I hope I will convince you that the two are related. So, the best example of that came from an experiment that we did many years ago, where we compared the responses of the aorta, a large blood vessel, of two strains of rats, the Wister Kyoto rat and the spontaneously hypertensive rat. And I don't think that I have talked about that already. Professor Kamoto, he's long gone now, a very distinguished professor in Kyoto, had looked in Japan, wanted to create a model of hypertension comparable to that observed in humans. In most humans, many people have a high blood pressure. Most people, we don't know why. I mean, I've said that already, but we then give it a nice name, and the doctor will tell you, you have essential hypertension. That means, sir, you have hypertension, and I have no idea why. But, essential hypertension sounds good, sounds scientific. Now, Professor Kamoto wanted to have a model of that, an animal model, because if you have a proper animal model, then you can start doing experiments and start trying to understand, how the disease progresses. You can try to find new drugs to affect the disease. It's very important to keep that in mind. I know that in your newspapers you read all kinds of things about animal research, but don't forget that medicine would not be what it is now without animal research and without proper animal models. So it's very important to do that. So he wanted to do that, and he was very bright and very patient. So he took, let's say, 100 Wister rats. That's one strain of rat. And he measured their blood pressure. And just as if I were to take the 24, 25 students in the room, if I would measure your blood pressure, I would assume that all of you are young enough to have a normal blood pressure, yet some of you would be a little bit lower, some of you would be a little bit higher, and the vast majority would be 120 millimeters of mercury. Okay? We would have a Gaussian distribution of blood pressure. And what Professor Chi-ming did is he started taking all the Worcester rats with the very low blood pressure and all the Worcester rats with the highest blood pressure. And he started mating them. And he did that for several generations, always taking the one with the highest blood pressure and the ones with the lowest blood pressure. And at the end, after many years and many generations, he ended up with a strain of rat that we still use 30 years later. A strain of rat where all the rats are hypertensive, become hypertensive. And that is what we call the spontaneously hypertensive rat, the SHR. And the Worcesters, that we're low in blood pressure, they will spontaneously never get hypertension. They can, even if they get very old for a rat, their blood pressure remains low. So basically, genetically, he separated the two groups. And thus we now have a model where we can compare the average person, average rat, with a rat with essential hypertension. Okay? And when we did that, and we looked first at the relaxations to acetylcholine, I was predicting that there would be less EDRF being released. Well, we saw something very interesting. At the beginning of the concentration, relaxation to increasing concentration of acetylcholine, at the beginning the two curves are identical. And up to about 50% of relaxation. But then this one takes off. And we get this difference in response. Now, because we knew about EDCF and endothelium-dependent contraction, we immediately said, well, this to us means that basically there is nothing different with the release of nitric oxide, but there is something else there. And the something else is, we hypothesized, EDCF. Thus, we took quiescent preparations, no pre-contraction, no gasoline, they're standing still in the organ chamber, and we gave increasing concentration of acetylcholine in rings of spontaneously hypertensive rats, or Worcester Kyoto rats, with or without endothelium. And this is a very easy slide to understand for students. There's nothing, and only in the aorta with endothelium of the spontaneously hypertensive rat do you see this very nice contraction, demonstrating that under those pathological conditions there is a big enlargement of EDCF mediated response. And this was the first time that we were forced to conclude that a pathological process can favor the occurrence of EDCF. And since this has been confirmed in a number of situations. For example, Professor Cohen, after he left my lab and went to Boston University, demonstrated basically the same with diabetes. This is an aorta of a normal rabbit, this is the aorta of a diabetic rabbit, the preparation from the normal rabbit relaxes nicely to acetylcholine. The preparation from the diabetic rabbit, it's very, very different indeed. And this is, as you can see, because here we have the release of EDCF. Okay, so those were two situations where EDCF started to become more important. Since those experiments, we have continued to explore that, and there are several situations where this is the case. Aging. Remember, aging is not a disease, it's a grace. But with aging, as we age, more EDCF is being produced, less NO is being produced. With obesity, I mentioned yesterday that there was less EDRF being released, but the most prominent thing is the occurrence of EDCF responses. How can we explain that? Well, if you look at a lean person, not me, I'm not lean, I'm too fat. If you look at the lean person, even the skinny students have fat tissue. Because that's a storage. That's in case you don't get dinner tonight, You want to have some storage of energy. And those cells are much more than simply storing energy. Those cells can produce substances that can go into the bloodstream and act at distance. Actually, those cells produce hormones. We can call them hormones. And people in obesity research tell us that fat is the largest endocrine gland of the body, especially in people like me. It's a very impressive endocrine gland. And those cells will produce two types of hormones. Good guys, and those we call adipokines. Adipo means fat, and kines, substance produced by fat cells. And some of those are very good. Adiponectin is a good guy. And others, like this one, lipocalin 2, is a bad guy. This inhibits inflammation, this favors inflammation. And as we go to obesity, the number and the size of the fat cells augments, and those fat cells, those fat tissues become inflamed and start to produce more and more of these and less of the good guys. You see? Okay, so you disrupt a balance. And as a consequence of that, obesity is accompanied by abnormal handling of lipids, insulin resistance, metabolic syndrome, and eventually diabetes. What I want to talk about is endothelial dysfunction that leads to atherosclerosis. And let me give you one example. I could talk for 2 hours on this topic. I'll just share one example. 2, actually. We worked, more recently in Hong Kong, we worked with lipocalin 2. And lipocalin 2, or we were interested in lipocalin 2. And lipocalin 2 is a pro-inflammatory protein. It is augmented in obese mice and humans. It is reduced by anti-diabetic drugs. And in humans, if you look at all the syndromes, the symptoms of abnormal symptoms that accompany obesity and lead to diabetes, lipocalin 2, the levels of lipocalin 2 are correlated to that. So what we did is we took mice, or we fabricated mice that cannot produce lipocalin-2. Lipocalin-2 knockout mice. Okay, good. And we took those mice and we followed them for a number of weeks. Two groups, one on a normal diet, one on a high-fat diet. Remember my American McDonald diet, you know? But now for the mouse. And this slide shows you changes in blood pressure. And if you look at blood pressure in the mouse from week 8 to week 22, you see that there is a tendency at the beginning in the young mice, the blood pressure increases and then stabilizes, stays the same. And there is absolutely no difference between the mouse that still can make lipocalin-2 and the mouse that has no lipocalin-2. Here we do the same thing, but we give the mice high fat. And as you can see, the young mice, wild type, that can make lipocalin, become hypertensive. And the absence of lipocalin for a long time protects against that. Now this is an important experiment because it shows that something secreted by fat can affect blood pressure. It also shows you, for those of us who work with genetically modified mice, that often you see nothing until you stress the system. So, you have no phenotype, the mouse looks absolutely normal, as long as it is on a normal diet. But if you stress it, then things go wrong. And then we looked at endothelium-dependent responses. But first let's look at the wild-type mice, at normal mice. They still make lipocalin. In very young mice, EDCF responses are very small. If the mouse becomes older, you see that progressively EDCF becomes more important. Illustrating what I said already, that aging is one of the factors that, permits expression of a greater EDCF response. But look here, those are young mice here. If the same age, but we've given them high-fat diet, you see that now there is a tremendous augmentation of the response. And if we compare that, I missed one slide. You'll take my word for it. You don't see this in mice that cannot make lipocalin-2. So this abnormal EDCF mediated responses is facilitated by the absence of lipocalin-2. Another thing that we did recently, remember on one of the schematics, I showed you that in adipose tissue that there is inflammation, and as a consequence of that, there is a abnormal secretion of adipokines, more lipocalin-2, number of other ones, less adiponectin. Well, we were interested also in that aspect, and we looked at the role of it doesn't matter. It's a cell membrane receptor. I want to show you how the system moves. I don't want you to remember all of that. So one of the key systems involved in inflammation is toll-like receptors. So what we did is we created mice. We took mice that are spontaneously, another genetic model, where all the mice get diabetes. We call that the DBDB mouse. And at the same time, we took a mouse and we knocked out this toll-like receptor 4. Again, I don't want you to remember the details of the manipulation. I just tell you, okay, here we have a mouse, and if I do nothing, it will become diabetic. and fat. Now, in the same mice, I now take out this receptor, TLR4. It doesn't matter to me. And I now compare the mice. And if you take the DBDB mouse that still has the receptor, it becomes very fat, and it becomes hypertensive, and it becomes diabetic if we measure blood glucose. And now I take my mouse where I've taken out the two, I have the knockout for the two aspects, the DBDB and the TLR4 receptor. And what do I see? I see that my mouse doesn't become hypertensive. I see that my mouse doesn't become diabetic. But if I look at the weight, it is much fatter. And I love this picture, because it shows you that's not because you are fat that things are wrong. It's because your fat becomes inflamed, because there's some inflammation in the cell. But fat per se, overweight per se, for what I'm talking about today, is not a problem. And of course I like that very much. To conclude that being a little fat is not necessarily bad for you. I think to me that's a very reassuring feeling. And this mouse I call the McDonald's mouse. She can go to McDonald's as much as she wants. She will not get into trouble. Good. Well, if we look at, this would be in the diabetic mice, the EDCF mediated response, and you see that we considerably knock it down by taking out this particular receptor. Illustrating again, I hope I've given you a few examples that pathology can have a profound impact on the occurrence of EDCF mediated responses. The next question, so we know endothelium-dependent contractions exist. We know EDCF exists. We know that it may play a role under physiological conditions, and an important role. We also know that in a number of pathological situations, it is exacerbated. It's bad, too bad. It's part of what we call endothelial dysfunction, more EDCF. Okay. What is EDCF is an obvious question. And again, we've tried to answer that over the years. And actually, I've shown you that slide already. Remember when we talked about arachidonic acid being transformed by cyclooxygenase into endoperoxide, and then with the action of thromboxane synthase producing thromboxane? And in the endothelial cells, we had a very similar thing, arachidonic acid, cyclooxygenase, endoperoxide, another enzyme, prostacyclin synthase, and prostacyclin. And you remember, prostacyclin was causing vasodilatation and inhibition of platelet aggregation. Okay? So, one of the very important pathways in endothelial cells is the metabolism of arachidonic acid by cyclooxygenase. So we obviously, when we looked and tried to figure out what EDCF was, we asked ourselves, how is arachidonic acid involved? Well, arachidonic acid, as I just said, is transformed by cyclooxygenase into endoperoxide, and then can go different ways, different prostaglandins. The ones that we will consider are those two. Prostacyclin, a good guy, and thromboxane, a bad guy. It's not a bad guy. It causes vasoconstriction, and whenever it is released, under physiological condition, it's a good guy too. It's only if too much of it is formed during pathology that it becomes a problem. Now, in this experiment, We're back on veins. This is a very old experiment, one of the first ones we did in the early 80s. We looked at endothelium-dependent contractions, and we repeated that in the presence of indomethacin. I think, have I mentioned indomethacin already? Indomethacin is a chemical that blocks cyclooxygenase. So if you give indomethacin, arachidonic acid cannot be transformed by cyclooxygenase anymore. Okay. So you stop the synthesis. And we stopped the production or the action of EDCF. And if we did that with other inhibitors of cyclooxygenase, aspirin, you know about aspirin, everybody knows about aspirin. Aspirin is an inhibitor of cyclooxygenase. It's the most popular one. If we use meclofenamate, the same thing. So from this, we quite early concluded that the EDCF must be a product of the metabolism of arachidonic acid. And when we then did the experiments comparing the WKY and the SHR, remember this would be the WKY, this is the SHR. Well, somewhere here you see the SHR here, blood vessel treated with indomethacin. It's gone. The abnormal response is gone. Demonstrating that also in this pathological model, the EDCF is a product of cyclooxygenase. And I must say, I still have to see endothelium-dependent contractions that are not explained by the production of one of the vasoconstrictor prostaglandins. To me, it's very clear EDCF is a metabolite of arachidonic acid. I will show you a little bit further which one I think is the most important, but theoretically, all can contribute. Again, there is no golden rule. In one blood vessel, it may be different. In a given blood vessel, with a given stimulus, it may be different. But let's take the straightforward path to show you how we proceeded. Okay, so in the 80s, we were convinced that everything was explained by cyclooxygenase. Then came a series of papers, among others by Sir John Vane, who got the Nobel Prize many years ago for discovering prostacyclin and thromboxane. And those people showed that, as often in nature, we start off with one protein, one receptor, or one enzyme, and then as we become smarter and our techniques improve, we discover that there are more forms, that it's not one cyclooxygenase. They came up, just as for nitric oxide synthase, you have one in the endothelium, one in the brain, there's several. So, isoforms, we call them. And those people concluded that there were two isoforms. Cyclooxygenase 1, COX-1, and cyclooxygenase 2, COX-2. And COX-1 was considered to be a housekeeping protein, was constitutively expressed in almost every cell, and was, in John Vane's thinking, the enzyme responsible for the production of the good guy, prostacyclin. But they discovered another isoform, COX-2, and that they discovered was inducible. When disease came up, when tissues became inflamed, when there was inflammation, then cyclooxygenase would come up and produce prostaglandins. And thus cyclooxygenase 2, COX-2, became, in their mind, associated with disease. So you had a good cyclooxygenase and you had a bad cyclooxygenase. And let's forget about all the pharmacology which this led to, the COX-2 inhibitors and all the problems with that. But I just want you to follow my thinking. So when we were confronted with two types of cyclooxygenase, and I knew that EDCF was being exacerbated, much stronger in disease, what did I guess would be the isoform? We said it must be COX-2, because that's the bad guy. Well, again, until you do the experiment, you don't know. And to our surprise, we were wrong in our hypothesis. In this experiment here, these are endothelium-dependent contractions in ring with endothelium, no contractions in the absence of endothelium. And I show you the effect of two drugs. This drug is a drug, it's not selective, but it is a preferential blocker of COX-2. So this should take out the bad guy. And this tinidab here is a preferential blocker of COX-1. According to my hypothesis, it should do nothing. But as you can see, it is the COX-1 inhibitor that takes out the response, while the COX-2 does not. And in the meantime, we have repeated that with more selective drugs. So pharmacologically, we concluded there must be a role for COX-1, which was very surprising and got me into trouble with my good friend Sir John Vane, because it didn't make sense. Now, a few years later, when I moved to Hong Kong, I worked with a very bright student, Eva Tang, who is now on staff, and if you come, you will meet her, or let's say when you come, whenever we get the paperwork sorted out. And what we did nowadays, if you do pharmacology, it's very hard to get your papers published in good journals. You need some molecular biology. So what we did, we got our hands on wild-type mice, mice where COX-2 was knocked out, and mice where COX-1 was knocked out. And we then compared the responses. And to make a long and beautiful study short, here is the bottom line. You see, in the wild type, endothelium-dependent contraction. In the COX-2 knockout, endothelium-dependent contraction. In the COX-1 knockout, no endothelium-dependent contraction. And this conclusion, the cyclooxygenase that is responsible is COX-1. Okay. Now, you probably haven't noticed, but so far I've made a very big jump, which I really shouldn't be allowed to do so. Because I told you endothelial cells produce EDCF, and this is blocked by cyclooxygenase inhibitors, and thus I assume that EDCF is a product of cyclooxygenase. But I don't know that. Because in our experiments, we didn't know where the COX is. It could very well be that the signal from the endothelial cell moves to the vascular smooth muscle cell and stimulates cyclooxygenase in the vascular smooth muscle cell. So we have not made the point yet, or I have not shown you the data yet, that allow me to conclude that COX-1 is located in the endothelial cells. I could show you bioassay studies. They are a little complicated because you can treat the parts of the sandwich with different things. But let me show you this experiment because it's the most convincing. Here what we did is we took SHR and WKY, 36 weeks old, when the hypertension is very well established, and we demonstrated that the COX-1 gene is overexpressed in the endothelial cells of the SHR compared to the WKY, but there is no difference in the smooth muscle. Just one experiment, and we have many more pieces of data that allow us to conclude that there is an overexpression of COX-1 in the endothelium, and that this is the reason why we have a greater release of EDCF. Now, very interestingly, again, this brings us in our thinking to a fundamental question in biology. When you take an animal model of a disease and you see a change, you always have to ask yourself the question, the old question, what comes first? Remember? I don't know when you do that in Korean, but to us, one of the key questions us when we are children is, what comes first? The chicken or the egg? You understand the dilemma? So here we see a difference in expression of an enzyme in hypertension. Is it a cause of the hypertension or is it a consequence of the hypertension? Okay? And when we tried to answer that, we took younger rats. At this stage, the rat is already hypertensive, but there is no overexpression of COX-1. If we wait longer, there is an overexpression. So what's your conclusion from that? What do you conclude? Is COX-1 something at the basis of the process or a consequence of the process? What do you think? If you compare those two. Cause or consequence? You can only two. Come on, use your brain. The animal has been hypertensive for a very short time. Nothing is abnormal from this point of view. If we wait longer enough, whoops, we give up. Cause or consequence? Say it! I know you know it. It's obvious, it's a consequence. If this were crucial in the hypertension process, we would be high up right there already. We're not. Illustrating, and if I were now to show you, I don't have the data, similar experiments looking at WKY rats, but getting progressively older, I would come to the same conclusion. As the rats age, this becomes more important. And this very experiment initiated my conclusion that the overexpression of COX-1 is not a genetic change in the SHR, but is a consequence of the fact that those blood vessels are for weeks and weeks exposed to a high blood pressure, and as a consequence, the endothelium ages more rapidly. So this is premature aging. Okay. But anyhow, it's in the endothelium and it generates those substances. So, of course, let me see what... Yeah. We have different candidates. Which one would it be? Which metabolite of arachidonic acid? Again, you've seen this slide, and there are many candidates. But over the years, certainly in the case of acetylcholine and the SHR aorta, we focused on those two. Now, intuitively, again, when we saw this response, and we concluded that it was cyclooxygenase that was creating the vasoconstrictor, which of those two was our candidate? But that's a vasodilator. Which one was the most logical candidate? Thromboxane. Because thromboxane we knew was a vasoconstrictor. So we say, okay, let's go and prove that thromboxane is the bad guy. So what we did, 20 years ago already, oh no, do I have it right? Yeah. We did this experiment. That is that we looked at those, endothelium-dependent contractions to acetylcholine under controlled condition and in preparations incubated. I don't know what this is, but this is probably molar or 10 to the minus 6 or 10 to the minus 4 molar. Something went wrong with the computer here. With dazoxibin, and dazoxibin is a substance that blocks thromboxane synthase. So, in those cells, cyclooxygenase still, let me get back a few slides. So, thromboxane synthase, so cyclooxygenase from arachidonic acid to endoperoxide, which by the way is a very labile substance. It's immediately transformed by other enzymes, thromboxane synthase to thromboxane A2. And dazoxibin, the drug that I just mentioned, blocks this enzyme. So all of this can go on, but no thromboxane. And as you see, to our surprise, again, logical, based on the accumulated knowledge, we formulated an hypothesis that seemed very logical to us. It has to be thromboxane. It's not. Does that mean that under other circumstances thromboxane cannot play a role? I'm not saying that. If we looked in the endothelium at the expression of those enzymes, now, there's 20 years difference between this slide and the one that I just showed you, just because we progress in thinking, as science is about. We progress in thinking. We can measure more refined things. We can do a better analysis. So we move on to the next step. And with Eva Tang again, we measured the expression of thromboxane synthase that transforms endoperoxide to thromboxane and prostacyclin synthase. And we found that those two enzymes are overexpressed in the endothelium cell. So theoretically, thromboxane could play a role, but in the case of acetylcholine, it does not. But this slide has a very important point. So, there were no difference in the receptors in the smooth muscle. I may come back to that later. What's the big difference between those two? Look, what do those graphs tell you? One thing when you do experiment is always to look at the graph and say, what do those cells, what do those tissues, what do they tell me? Now, if I look at this, I say, well, prostacyclin and thromboxane, the same thing. They're both augmented. But what's the difference between the two? The scale. This is an incredible presence of prostacyclin synthase and an incredible overexpression. You agree? So that message came across to us. Why on earth? What would that mean? Prostacyclin is a good guy. It's a vasodilator. It doesn't make sense. When Eva Tang showed me that, I said, it doesn't make sense. So, over the years, we tried to figure out what was being released. And one of the first things we did is we measured the production of endoperoxide, which is the precursor. And sure enough, if we measured that, this would be WKY aorta. Actually, no significant release of endoperoxide. But in the SHR aorta, if we gave acetylcholine, we had a very large increase in the production of endoperoxide, and this was abolished by removal of the endothelium or by our good old indomethacin. So, clearly endoperoxides are key somewhere. And for several years we felt, I actually was convinced that it's not thromboxane. Conceptually, it cannot be prostacyclin, because prostacyclin is a good guy. So it has to be endoperoxide. And I wrote several papers and I went to many conferences saying that. More recently, in Paris and in Hong Kong, we revisited that. And we measured the release of prostacyclin in aortas, controlled or stimulated with acetylcholine. And we got a massive release of prostacyclin. A massive release of prostacyclin, which is prevented by indomethacin. Remember this drug, the preferential COX-2, there's a little inhibition but not much. But if we use a much more selective COX-1 inhibitor, it's gone. So we were confronted with the massive release of prostacyclin, a massive overexpression of prostacyclin, and thus we had to start thinking that thromboxane with acetylcholine not involved, the other prostaglandins, I didn't show you the data, not involved, endoperoxide could be prostacyclin a candidate. We'll come back to that in a moment. We were thinking about those two, and as you probably know, we've said that when a cell secretes a substance that go and changes the activity of another cell, it usually will interact with cell membrane receptors, proteins on the cell membrane of the next cell that will then be connected. We've seen examples of that. And the two receptors, we looked at all of those. They don't play a role, so let's not waste time on that. The two key receptors that could potentially mediate responses to prostanoids in vascular smooth muscle are thromboxane, receptors for thromboxane, or receptor for prostacyclin, vasoconstriction, vasodilatation. When we look at the two types of receptors, the presence, the expression, mRNA, the expression of the genes for those receptors, there is actually no difference between thromboxane receptors in expression in WKY, in the normotensive and hypertensive rat. Neither is there one for prostacyclin receptors. So that's not where the answer comes from. We know that I've told you already that the thromboxane receptor, thromboxane itself is not involved because this substance does not affect it. We know that this substance may contribute, endoperoxide, and we now are beginning to ask ourselves, what about prostacyclin? Now, if we look at the different receptors that could be involved, the one that can mediate contraction in vascular smooth muscle is the receptor for thromboxane, which abbreviated is thromboxane prostanoid receptor. So when we ask ourselves the logical question, which receptor is involved, we had to say, okay, it must be the TP receptor. And indeed, already 20 and more years ago, when we used substances like this one, now this is much more recent, but substances like this one, we could see this would be contractions to acetylcholine in the SHR aorta, endothelium dependent, abolished by a TP receptor blocker. So very clearly, the receptor involved in the response is a thromboxane prostanoid receptor. Yet, as I showed you, there is no difference in the amount or expression of that receptor between the two aortas. Okay, but so if we measure the receptor in the aorta of the WKY and the SHR, same amount of receptors. So from a molecular biologist point of view, no difference. But this is where pharmacology comes. And so it's not because you have the same amount of protein that you recognize with an antibody, if you do, or a gene expression. That doesn't mean that the receptor works in the same way. So we did experiments, and we looked at the effects of increasing concentrations of endoperoxide. When we did this experiment, endoperoxide was a primary suspect. And you can see that there is a slight contraction in the aorta of the WKY, but that this is very hyper-responsive in the SHR. So although we have the same expression and the same number of receptors, in the SHR they are much more responsive. What would be the next question that we would ask ourselves here? Now we see a difference. Just as we saw a difference with expression of cyclooxygenase. We don't see a difference in expression of TP receptors, but a difference in sensitivity. What is the next question that we would ask here? As we did before. Chicken or egg? Cause or consequence? So we took the same approach as for cyclooxygenase. We took very young rats, barely hypertensive, older, adult, well-established hypertension, and in the young animals, at a time where cyclooxygenase is not overexpressed, the receptor is already there, abnormally sensitive. So whereas, in my thinking at least, the overexpression of cyclooxygenase is a consequence of the hypertension, this may very well be a genetic platform on which the endothelial dysfunction can express itself. You understand the difference between the two? Okay, so that's one thing. So now we find something at the level of the TP receptor. Can it explain this very large contraction? Well, although I showed you the data, I told you endoperoxide, but to be honest, I never say that in public, and certainly not in those years. This is an increase. It's statistically significant, but those are not very large amounts. Nothing compared to what we see here. See with prostacyclin. So somehow we still had, after we found this and this, we still had to figure out what the role of prostacyclin is. And here we compare in the WKY without endothelium. Let's make it simple. Don't look at all the curves. I just want to show you that in the WKY, prostacyclin is at 12 weeks old. Prostacyclin causes a very good relaxation. Prostacyclin is a good guy, is a vasodilator. Okay. Let's now look what happens with the WKY. You see the difference? No relaxation. So there are two receptor changes in the SHR. One, the TP receptor becomes better, constricting one, and the IP receptor doesn't respond anymore. So whatever amount of prostacyclin we generate, it cannot cause activation of the TP receptor, of the IP receptor. And as a matter of fact, if we give enough prostacyclin, it eventually will cause contraction. which we can block with a TP receptor blocker. Prostacyclin at high concentration can activate TP receptor. And now I think I understand what goes on. Under normal circumstances, in normal tension, if we give acetylcholine, we will cause a release of PGI2, that's prostacyclin, that will get to IP receptors, cause the production of cyclic G, AMP, and relaxation. And not enough prostacyclin is there to even touch TP receptor. In the case, and this is a very complex schematic, don't try to understand it all. In the case of hypertension, now the IP receptor doesn't respond. And something we still are working on at the present time. I'm convinced, I believe, I hope I will be able to prove it before I retire or die. I think that when you remember what I told you about presynaptic inhibition, prejunctional inhibition, where noradrenaline was being released and then did feedback on the nerve ending so that there would be no exaggerated release of noradrenaline. I think the same operates here. You know, normally the endothelial cells secrete prostacyclin that goes to the IP receptor, causes relaxation. There must be also IP receptors on the endothelial cell so that if the levels of prostacyclin becomes too big, that we switch this on. And I think that that is missing in hypertension. And again, it's a working hypothesis we're trying to prove now. So just me dreaming here, you know. All I've shown you so far is data. Now this is me extrapolating. And I believe that as a consequence of that, the fact that this feedback does not exist, that we generate more and more prostacyclin. We overexpress. The system feels that it doesn't get the good signal. And as a consequence, it goes wild and produces more and more prostacyclin to the extent that prostacyclin now can activate TP receptor and cause endothelium-dependent contraction. And this then illustrates the fact that a good guy can become a bad guy. And that is the title of one of my papers. When a good guy turns bad, or when a good guy turns ugly. And I mean prostacyclin, which normally is a protective molecule, but if we interrupt the normal control, it come more and more, and we go to the point where then those concentrations, remember the very high concentrations that we measured, remember the extremely abundant expression of prostacyclin synthase. The system is geared to produce vast amounts of prostacyclin that cannot do what it should do, but unfortunately, now at those concentrations, causes contraction. Okay, what other questions have we asked ourselves over the years? How is EDCF released? What causes it? Well, that's easy. I mean, if by now you don't know that in most cells that are being activated, what is the activator signal? Who knows? Eventually, in the cell, what's the ion that will activate the system? No. In the cell, acetylcholine hits the cell membrane. The nerve impulse comes. What happens? What increases in the cell? What ion concentration increases? Which one? What's the activator ion I'm talking about? Calcium. So it's a matter of calcium. And we can mimic that by using this substance. This substance is a calcium ionophore. It makes holes in the cell membrane so that calcium can flow from the inside the cell. And if you do that, you see that you get this is a very old experiment, older than most of you. But we could see that it causes endothelial dependent contractions. So calcium is key. And nowadays we can be a little bit more sophisticated and measure that. In this experiment, what we do, shoot, I forgot again. Release. There's a spelling mistake in my slide. I hate that. So what we do here, the green color, it's a fluorescence technique by confocal microscopy, and we can measure if the cells become green, that means there is more calcium in their cytosol. Okay, that's all you need to know about the technique. And this is before, and then when we give acetylcholine or the calcium ionophore. And this is the WKY, this is the SHR, WK. And if we use acetylcholine and forget about all of those things, we see that systematically acetylcholine causes a greater calcium release in the SHR than in the WKY. And this illustrates something that we don't understand yet, which is that somehow the handling of calcium by those cells is abnormal. Now, it has been done for 50 years, but in hypertension, abnormal handling of calcium at all levels is a problem. Okay. If we give the calcium ionophore, and that's why I wanted you to look at those slides, what do you see about the signal? Do you see that this is basically the same? Whatever the SHR that, oh, my refresher coffee. Thank you very much. Thank you. God, they treat me so well here in Zurich. You see? It's the same. How do you explain that? Do you have an explanation for that? Is that logical? Or is that strange? This difference between acetylcholine and... Can you understand why this is actually not surprising? I would have predicted that. Why? Well, what did I tell you? What does the ionophore do? 823187, calcium ionophore. I told you a minute ago. What does it do? It makes holes in the cell membrane and calcium can flow in. Is there any reason that this would be different in the WKY and the SHR? You make pores. Come on, physicists. There's absolutely no reason that this would be different in the SHR and in the WKY. You know, basically 823187 is a detergent. It interrupts the cell membrane and it allows calcium to flow in. This has to be the same in the two. And tissues don't lie. They show me that. And actually, I don't show you that, but if we look at endothelium-dependent contraction, the calcium ionophore is practically as good in the SHR as in the WKY. To cause them, they're smaller in the WKY because they don't have this hyper-responsiveness of the TP receptor. Okay. So, calcium is key. What does calcium do? Well, in the case of eNOS, of endothelium nanosynthetase, calcium, that's a calcium-sensitive enzyme. Certain enzymes work better in function of the calcium concentration, the pH, etc. But they're catalysts. Now, cyclooxygenase, everybody knows that, is a calcium-independent enzyme. You give it arachidonic acid, it will transform it, irrespective of the calcium concentration. So the calcium dependency comes from something else. And that something else is here. At the level of, I think I had that on one of my schematics. Arachidonic acid produced that will feed into COX-1 and give that is liberated from membrane lipids through an enzyme called phospholipase A2. And that enzyme is calcium sensitive. So if the calcium goes up, more arachidonic acid is produced. And then we can go down the cascade. So acetylcholine activates its receptor, leads to an increase in calcium. This activates phospholipase A2 with a greater production of arachidonic acid that can feed into cyclooxygenase 1 and produce EDCF. And indeed, if we use indomethacin or meclofenamide, I've already told you that, we get rid of endothelium-dependent contractions. If we use an inhibitor of phospholipase A2, we get rid of endothelium-dependent contractions. So we have a pretty good idea of what happens here. So, stimuli at the cell membrane or direct holes in the cell membrane by A23187 increases the calcium concentrations. This activates phospholipase A2 that transforms membrane lipids into arachidonic acid that then can feed into COX-1, forms endoperoxides, and which then can be either directly activate the TP receptor or be transformed in thromboxane, but that's not likely to be the case for acetylcholine or prostacyclin to cause the endothelium-dependent contraction. Do free radicals contribute? Well, free radicals contribute. Now, do you know what free radicals here? Oxygen-derived free radicals, superoxide anion, H2O2. I'm sure you've heard of those things. Well, they modulate, they play a role. If we measure them again, we can see that they increase during endothelium-dependent contractions. And we feel, do I have slides? No. And we feel that those free radicals amplify the action of COX-1 both in the endothelial cells and the smooth muscle cells. But that's really too complicated for you guys, so I'm going to not talk about it. I just want you to remember in the back of your brain that free radicals can contribute, and that if there are more free radicals, endothelium-dependent contractions are bigger. And this is, for example, the reason why in diabetes, because diabetes is a disease characterized by increased oxidative stress. That's why, one of the reasons why EDCF responses are bigger in diabetes. But I really don't want you to go into that. The last aspect that I would like to cover is, okay, I've now for an hour and a half been talking about EDCF, and I haven't mentioned nitric oxide and the endothelium-derived relaxing factor. So we have a cell that can produce NO and EDCF. So it was very logical at one time to ask ourselves the question, how does that work? Do they work together or against each other? Or, you have an acceleration, you have a brake. How does it work? So we tried to do that. And to do that, we did the following experiments. Here you have to listen well. So I hope I've convinced you that when we give acetylcholine to an endothelial cells, two things can happen. We can activate NO synthase with the production of NO and relaxation to cyclic GMP. Or we can activate cyclooxygenase. Well, actually, we don't activate cyclooxygenase, we activate phospholipase A2 and other things. And we produce more EDCF that activates TP receptors. At one time, I was wondering how those could interact. So we used 3 different drugs. One drug is an inhibitor of NO synthase. The other drug, I've already mentioned it, I think, is oxyhemoglobin. Oxyhemoglobin is a very large molecule that cannot get inside the cells, but it can get between the cells. And between the cells it will scavenge, it will take up nitric oxide and bind it to its heme groups. It's a scavenger of NO. And methylene blue I've already mentioned. That's an inhibitor of soluble guanylate cyclase. It blocks the production of cyclic GMP and the effect of NO. And we looked at the effect of those 3 drugs on EDCF-mediated responses. And we figured if they are affected by NO synthase, that involves the role of NO. If they are affected by oxyhemoglobin, that involves an interaction between the two on the way from the endothelial cells to the smooth muscle. If they are affected by methylene blue, that means that NO and EDCF work on the smooth muscle. Yeah. Very primitive, simple thinking. So we did the experiments. And here would be the response in the absence of any drug, and that response was greatly augmented by oxyhemoglobin and by the inhibitor of NO synthase. Now, to be honest with you, most of the slides that I've shown you today were in the presence of an inhibitor of NO synthase, because it gives such nice EDCF-mediated response. So it's easier for us to do our analysis. But to our surprise, methylene blue did not work as the other 2. Okay? So our interpretation was that LNMMA augments. That means that NO is important in cutting down the production or the action of EDCF. Oxyhemoglobin augments to the same extent as LNA. So somehow the interaction is not in the endothelial cell, because otherwise oxyhemoglobin would not do anything. But the interaction is not at the level of the smooth muscle, because methylene blue does not have the same effect as the other 2. Okay? Thus, I concluded, and I went around the world telling that story, that NO interacts chemically with EDCF on the way between the endothelial cells and the smooth muscle cells. Got a lot of mileage out of that. And this is wrong. It shows you that when you do experiments in pharmacology, you never trust a single drug, and you never believe selective or specific. Doesn't exist. You have preferential, but never really selective. And indeed, I went around the world telling the story until I came across that paper. 1926, in a very good journal, demonstrating that methylene blue is actually a muscarinic antagonist. For years we've used it to inhibit cyclic GMP production, and actually, honestly, most of the effects of methylene blue in early experiments were not due to inhibition of cyclic GMP production, but at least when we were working with acetylcholine, but working because it worked like an inhibitor of acetylcholine. And to me, this is one of the most surprising or most painful lessons in humility in my career. It also reinforces my conviction that I have to tell young people who work with me, go to the library, look what people have published. Never think you are the first. Somebody before you may have done something that is crucial. Nowadays, young people, I don't know, you're in bioengineering, but in medical research, they go to the computer, they go to PubMed, they look 2 or 3 years, finished. And they ignore everything that has been published before. And that can be fatal. Now, the saving grace is that we discover this. We realized ourselves that I had been stupid. So I didn't have somebody else telling it on the stage, Vanhoutte is wrong. I could say it myself, which is a lot better, of course. Less painful than if one of your competitors tells you that. And here, using other drugs, it doesn't matter. We found that it is exactly the same thing. So our conclusion then was that I know inhibits EDCF, but most likely, simply, at the level, under acute conditions, at the level of the vascular smooth muscle cells. And then came Eva Tang. Now this is, you probably don't know that word, so maybe I shouldn't show that slide, but this is one of my favorite slides, and it tells us about the journey and the voyages of the 3 princes of Serendip. Now, who knows what Serendip is? Serendipity is. Serendip is the old name for Sri Lanka. Old name. And the 3 princes of Sri Lanka were going through Persia. This is a French story, but based on a Persian story. And the 3 princes were lucky. Everything they found was by luck, by chance. And from this very book, the term serendipity was coined at the end of the 18th century in England to describe luck. And I always insist when I talk to young people, especially the beginning of a career in sciences, sometimes you hear people say, oh, Professor Dongmei-Wu is lucky. I am not lucky. Or Vanhoutte has been lucky. Of course I've been lucky. Everybody is lucky. But most people don't recognize it. And one of the biggest dangers in science is to have an hypothesis and to find data that are not fitting with your hypothesis, and then to try to get around it, not listening to the message. And sometimes a message comes out of the blue, serendipity, and one is often tempted to ignore it. And particularly young students, if you work with somebody like me, and I say, well, I think that this is going to happen, and as a young student, you find something else, especially in Asia. It's very difficult for the student to come and say, Vanhoutte, you're wrong. And this is something that you have to learn to do. And this is what Eva Tang did. Because when I moved to Hong Kong, I asked Eva Tang to start activating the EDCF program again in Hong Kong. So I told her, you take a ring, you contract it with something, you check whether the endothelial cells are there, you incubate with LNA, you do a dose response curve to acetylcholine, you will have EDCF responses. And Eva Tang tried that, and then she stormed one day in my office and she said, Prof, EDCF does not exist. And I say, Eva, EDCF existed in Antwerp, Belgium, my home country, existed in Rochester, Minnesota at the Mayo Clinic, existed in Houston, Texas at Baylor College of Medicine, existed in Suresnes in my lab in Paris. Don't tell me it doesn't exist in Hong Kong. It doesn't make sense. And this brings another concept, message for you is that you will do experiments with your supervisor, and you will come to an experimental finding, And you will then sometimes say, hey, this is different from what is published. And then there are two ways you can say. You can say, well, I'm right, and the others are wrong. Or if it's different from what it is published, and your supervisor expects you to find what is published, you put it aside and you forget it. That's often the case, because you don't want to go into his or her office and say, you're wrong. I did the experiment. But I've learned that most investigators are honest people. If you're not honest, you don't go into research. I mean, it's not the way to become rich, I can guarantee you. So, most investigators are honest. And if you find a difference, or if you don't find a difference, it is simply, usually, 99% of the case, because you don't do the same experiment. So I sat down with Eva Tang, and we discussed, but how do you do this experiment? And usually, in our experiment, we do a potassium contraction to get a reference contraction, then we do a constriction, then we give a quick shot of something, see whether or not there is a decent relaxation, wash out and go about our business. But this is not an actual trace, this is better. But she did a full dose response curve to acetylcholine, which lasted about an hour and ended up with very high concentrations of acetylcholine. So with other words, in our viability test, she was maximally stimulating the endothelial cell. And we could assume with a very large production of nitric oxide. So I told her, we agreed. I say I told her, but I guess we agreed together. Why don't we skip this part? And if we skip that, we saw the most beautiful EDCF responses that you could imagine. And this brought us to the concept, here, we release NO here. Now, NO is a gas. It's gone within seconds. And then we wash it away. We wait for 1 or 2 hours for, repeated washing. There is not a trace of NO left in the system. Yet, no response. This brought me to the conviction that NO released by the endothelium exerts a long-term inhibition on EDCF responses. And we could demonstrate that by, this would be no pre-exposure to acetylcholine. If we used exogenous NO, we had exactly the same thing. And this is at the level of the endothelium, because the response of the TP receptor is not at all affected. And thus, we now conclude that, this is not the best schematic for that, let me skip that. We now conclude, I'm now convinced, that if we have sufficient NO produced by the endothelium, that there will be repression of EDCF-mediated response. If the NO starts to fall down, EDCF can raise its ugly head. And this then brings the two together. An endothelial dysfunction is a reduction in NO release, permitting an emergence of EDCF-mediated response. And I could show you many data now that we do now with mice and genetic models demonstrating that. So remember, if we think about disease and endothelial dysfunction, we have the two aspects. Lesser NO, more EDCF, but in most cases the two are related. As long as your endothelium produces enough NO, you're safe. You're okay. Okay, final point. Is this only a rat and mouse story? Or is this important for you and me? And the answer to that came from Professor Tadei, already quite a few years ago. he worked in my lab and he became interested in EDCF and endothelium, and then went back to the city of Pisa, you know, where the tower is skewed, and he did this experiment. And here he measured now forearm blood flow in humans, and he looks and he has a needle in the artery, so he can infuse increasing concentrations of acetylcholine. And he does that in normotensive people and in essential hypertensive subjects. And you see that there is a remarkable reduction in vasodilatation to acetylcholine in the forearm of people with a high blood pressure. Untreated, no medication. But the beauty of his experiment, and people knew that, you know, some guy described that years before. But what he also could do, since he had a needle in the brachial artery, he could infuse indomethacin. And he did that. And if he repeated that, the response was almost normal. So with the local infusion of indomethacin, he normalized, basically, the endothelium-dependent vasodilatation to acetylcholine. This is exactly what I showed you in the aorta of the SHR, you know, before and after indomethacin. Normalization of the response. And this is very, to me, indirect, but in humans it's always indirect. It's so difficult to do human research from that point of view. It is very conclusive. He did something else. He took normotensive subjects, an essential hypertensive subject, and separated them in age groups. And then infused indomethacin to see the increase in blood flow, which he can attribute to EDCF, withdrawal of EDCF. And he also infused L-arginine. Do you remember what L-arginine is? Long time ago. What's L-arginine? Who remembers? Remember NO synthase, L-arginine to NO to citrulline. It's the substrate for nitric oxide synthase. And he gives that on the proposition that by giving L-arginine, he will make the NO system more efficient. If you look at normotensive subjects, L-arginine, up to, you know, relatively, I wouldn't say elderly, this is not elderly, I'm much older than 60, so that's, you're not old at 60, you're still very young. You see that there is, L-arginine works well. It works better and better. That means that as we get aging, to give a little arginine is more helpful than when you're young. But you see, there is relatively little impact. And we interpret that as demonstration that the NO system generation, the generating system, is still functioning. And there is very little EDCF. If you look at essential hypertensive patients, at the beginning everything is good, but as they become age, their NO system goes downhill, and the EDCF system becomes more and more prominent. And thus this brings in another thing that we talked about, age coming in here and here. You know. This fact that the predominance of EDCF-mediated responses is a sign, in part, of premature aging of the blood vessel wall. And thus I do believe that all that work that we did over the last 30 years, looking at, you know, animal blood vessels and EDCF-mediated responses, that this is highly relevant to the endothelial dysfunction in humans, and that it can explain a lot of the situations that we have encountered in the clinic. That's it. I'm done for this year, or this semester. Thank you very much for those of you who attended. I appreciate it.